Welcome. Today we have gathered a very distinguished panel to speak about the future of therapeutic and vaccines. And this time with a particular focus how novel modalities can influence the opportunity to advance new medical solutions, breakthrough solutions for patients, whether as drugs for therapeutics or vaccines for prevention. And I'm very pleased to introduce to you the colleagues in this panel. Penny Heaton, MD and leader of the Bill and Melinda Gates Medical Research Institute, CO. Mads Krogsgaard, Thomsen, PhD and Chief Scientific Officer for Novo Nordisk. John Maraganore, PhD, CO of Alnylam. Jeff Spaeder, MD, Global CMO and CSO for IQVIAGeorge. George Scangos, PhD, CO of VIR Biotechnology Incorporated. Before we do a bit of an introduction on this particular opportunity of new modalities, I think the organizers wanted to have introduce us to the poll function. So please participate actively in giving us your view. Here it was simple, it was just yes or no, as various questions will be raised to gather understanding what you as scientists, business leaders, colleagues think about the direction of various healthcare related questions. While you have another few seconds to review and send in your contribution on this poll, please also during our panel raise questions that you want us to ask after we've had each of the panelists share some of their reflections on the topic. As an introduction, I wanted to say that we have seen many advances in broad aspect of healthcare to come in the intersection of good biomedical science, new technologies that cover breadth from enabling technologies to give insights into disease to technologies that we focus on today in designing modalities for treatment or vaccination. And more recently, also digital becoming a very powerful additional contributor into science, technology, and digital. As we think about modalities in particular, we have traditionally viewed them as small molecules, orally usually available pills, biologicals, and more recently dominated by antibodies, nucleic acid based that we will hear colleagues here being experts speaking about, viral vectors. Actually, we heard earlier about cell therapies from Dr. June, and vaccine seems to embrace many of these options, particularly traditionally protein-based vaccines, more recently also nucleic acid-based or viral-based. And to just close out my introduction, while many of these modalities have introduced huge leaps in innovation across so many diseases that we have shared over several years of pre-Galien panels, in these particular times, I thought I'll just reflect on how they have contributed in a concerted response to COVID. Small molecules, many of them quickly repurposed from viral diseases, have been in the first line of response to COVID, and you're aware of remdesivir from colleagues at Galien that now is authorized to be used in treatment of SARS-CoV-2. And many other therapies in polymerase and protease inhibitors. Our company Pfizer has, with many other great companies, participated in clinical studies that tries to advance opportunities for combination therapies. Biologicals has also been part of the very early response. We heard earlier from George at Regeneron speak about what he's doing. We're also aware of efforts by Lilly to raise monoclonal antibodies, either single but likely more preferable in combination to combat resistance. And oligonucleotide, we will learn, are even explored in very innovative ways, including unique delivery system. And actually, two of the panelists, George and John, are collaborating in this space. When it comes to vaccines, I think we have more than 150 programs from preclinical to clinical development. Never before has it been such a crowded field, which should bring optimism that even with the most devastating attrition, we should have great successful vaccines. And we are exploring new technologies such as mRNA, where I think colleagues at Moderna spoke earlier. Moderna and Pfizer with BioNTech are, I think, the most advanced in the mRNA area of COVID-19 vaccines. Also here, viral-based, I saw my colleague Paul Stoffel speak about that too from J&J, and we've heard about the Oxford AstraZeneca effort using adenoviral delivery. And we have the traditional and very proven protein-based from companies such as Sanofi Glaxo in a unique partnership, and also Novavax. So it's really great to see that the response against COVID, it's less than 10 months old. We actually have mobilized several of the existing, rather now established modalities and several new. So it becomes kind of a great example how technology and science come together. And I think there is a need for much more digital effort. We are overwhelmed with the increasing knowledge base about COVID-19 publication. From being completely absent until December 2019, most journals have between 20 to 70% of the papers in recent months being about SARS-CoV-2, COVID-19. And digital solutions, and maybe Jeff can speak about that, will be extremely helpful in fully giving us insights to this tremendous flow of information about COVID-19. And obviously also will be key as treatment of vaccines hopefully will be rolled out, ability to follow patients, how they perform, and gather basically in real time experience using digital health-based tool, how various patient types can do better on one or another type of treatment intervention. Now, many of the first efforts have been focused on clinical trials in typical territories of US, Europe, and China, and a few other Asian countries. And it's great to have Penny Heaton here representing, maybe in her role at Bill and Melinda Gates, one of the biggest organizations that are making an impact to think about global health, and listen to her thoughts about not just how we can advance the super tech technologies we are designing for COVID-19 quickly to a global access, but also think about many of these technologies that we will hear about today, how you can advise us to find ways to distribute them, how to deliver them or simplify them to be over a few years equally present and available in Africa as in Boston, San Francisco, Shanghai. With that opening remark, I will now turn to my great colleagues in the panel and ask each of you to share some insights, how you have advanced science and technology particularly, and please feel very welcome to also think about how you integrate digital health, but with a particular emphasis how you have over the last few years evolved modalities as being the key to unlock promise for healthcare. So I'll start with Mats Krogsgaard. And I know, Mats, your company actually started as a very profound small molecule company. Under your leadership, you focused on peptide proteins and have done a tremendous success. And I think, you know, even exploring additional technologies that allow us to think about everything from metabolism to aging. Please, Mats.
during my tenure, 20 years now as CSO and head of R&D at Novo Nordisk, I think we've come to realize that where it all started back 100 years ago with our company based on the discovery of insulin at University of Toronto, we started with biological medicines, basically replacing therapeutic or proteins that were lost due to type 1 diabetes, growth hormone deficiencies or whatever, into evolving into a company that also tried to actually go into the small molecule space like much of Big Pharma has done. We have, however, come to realize that the real raison d'etre for our company is actually to continue to evolve biological medicines, and when we failed ultimately with the inhaled insulin, because the holy grail of giving insulin back to people is somehow to make it available in a non-invasive manner, just like Fred Banting, the Nobel laureate, tried to do with one of his student mates who had type 1 diabetes back in 1922. When that failed, the inhaled insulin, we moved into the oral space to say, okay, now we'll take the toughest route of all, namely to try to make oral biological or protein application possible, and that's where we have succeeded today. And now, of course, we are trying to combine it with digital medicine and actually use a lot of the approaches that all of us in the industry are to try to make drug adherence and compliance and ultimately patient outcomes better.
That's great to hear, and it's been very impressive how you have pioneered to turn peptides like your GLP-1 into orally available, and that opens up, I think, completely new space how to think about peptides. John Maraganore, heading Alnylam, have been a true leader in developing RNAi, and I think we have collectively together with you developed new solutions for amyloid TTR patients. So that's great to see. And I know you also are thinking about how RNA can play a role in COVID for therapeutic for the future. So please share with us a little bit your journey and vision for the powerful RNA in as a new therapeutic modality.
Yeah, thank you, Mikael. I mean, when I joined Alnylam 18 years ago in 2002, RNAi was really a lot of promise and hope at that point in time. There was no animal data, no in vivo ability to demonstrate, or no demonstration that you can harness this natural cellular mechanism. And so when we started at Alnylam, we knew that we had to conquer some of the key technical hurdles of advancing RNAi therapeutics as a new modality for human health. And it's been quite the journey, as you probably know, where there were many moments in which the elation turned into exasperation over time, but we stuck with the science. And over the years now, we've really demonstrated the ability of harnessing the RNAi pathway in cells to target disease genes and achieve a robust and remarkably durable silencing of the target gene with our small interfering RNAs. We're very proud to have brought 2 products to market at this point in time. One, as you know, is in the space where we're trying to help patients with hereditary ATTR amyloidosis with polyneuropathy in our case. And we also have 2 products that we expect to have approved by the end of this year, including one in collaboration with Novartis for the treatment of hypercholesterolemia with a biannual dosing regimen. So the technology is really now positioned to not only address the genetic medicine opportunities that we started with in the rare disease space, but we're also poised to now address very common diseases with a very differentiated mechanism of action that we think can be transformative for the management of a wide range of human disease.
Thank you very much. And I think it really opened our eyes when it comes to knocking down at the mRNA level. We then translate to drastically reducing or removing proteins, and you can see really intriguing biological outcomes, which may be sometimes quite different from what we've seen by antagonizing protein function with a small molecule. So a lot of really pioneer insight to the human genes and their role in disease by that technology. George, as a CEO of VIR Biotechnology, as the name indicates, you have taken as one of your missions to revitalize our treatment options in virology. And I think you also have a particular internal focus on the use of antibodies, while it's pretty impressive to see how you try to incorporate partnership to broaden the impact you can have in virology beyond what you design internally. So I'm eager to hear your thinking on what brought this new revitalization with VIR as a pioneer into this area.
VIR is about 4 years old now. We started completely with a focus on infectious diseases at a time when that wasn't a fashionable thing to do, but we saw huge unmet clinical needs, even in things like flu, where some number of tens of thousands of people die every year in the US from flu, hepatitis B, a number of other diseases. So we assembled a collection of technologies and programs. We have I think now a leading program on hepatitis B. That's a collaboration with Alnylam. One of our lead molecules there is an siRNA that is generating, I think, quite compelling data in clinical trials. We have a program for what we think will be a better way to protect people from flu. We have a collaboration with the Gates Foundation around using CMV to elicit T-cell responses to inserts that you can insert into there. It's a very potent T-cell inducer, and that's focused on HIV and TB vaccines initially. And we were going happily along, and then came COVID, right? And so we jumped in with both feet, and we're taking many approaches now to COVID. We do have antibodies we're bringing forward. Our approach has been a little bit different from what you heard about from Lilly and Regeneron. You know that antibodies can neutralize viruses, and you know that viruses can readily mutate to escape inhibition by the antibodies. So the approach that Lilly, Regeneron, and AZ have taken is to combine antibodies, have a cocktail of two. And the hypothesis there is that two antibodies will provide a sufficient barrier to the development of resistance, even though resistance develops fairly readily to each of them alone. We'll see if that bears out over time. We've taken a different approach. We, like everybody else, jumped in and isolated antibodies from COVID recovered patients. We also looked in patients who recovered from SARS, and the rationale being that if we could find an antibody generated in response to SARS that also potently neutralized COVID, that it must bind some epitope that's difficult for the virus to mutate, since it will have been conserved through all those years of evolution. We did find one. That's our lead antibody that we have moving into the clinic. If you look at the 100-some-odd thousand isolates that have been sequenced now of COVID, the mutations in that region are very rare, only a handful of them. We can isolate resistant mutants in the lab. We can't force them. So we're optimistic that that single antibody will provide a barrier to resistance that's equal to or better than the combination of the antibodies that are directed to a different region. We paid a lot of attention to effector function. This is an emerging area of understanding of how antibodies work. Most antibodies have some effector function, but it's a rheostat. Some have much stronger function than others. It has to do with the off rate of the antibody. It has to do with the angle at which the antibody binds to the target, and whether the FC region is sticking out and accessible or kind of buried on the surface. So we paid particular attention to having an antibody with potent effector function. That effector function can be enhanced. Some very interesting work from Jeff Ravitch's lab at Rockefeller about how to enhance effector function. It can also be eliminated, so you can engineer the effector function to have the properties that you want. And of course, half-life is important, especially if one thinks about using the antibodies prophylactically as well as therapeutically. You'd like them to be nicer to administer them once every few months rather than monthly. And so we've extended the half-life. So our antibodies are a little different from the other antibodies moving forward. It's in phase 3. We'll have data very soon, and we'll see. We're working with GSK, one of the partners that we've signed up on the antibody, but also in functional genomics. We've done CRISPR screening to identify cellular targets that are whose inhibition prevents the replication of COVID and other respiratory viruses. If you pool the data you get from the various viruses, you get very complex patterns, and it takes actually, I think, to get back to one of your questions, relatively sophisticated machine learning algorithms to make sense of all of this. You can put then these hits that you get out of the screen into pathways. And then, of course, you can identify molecules that intervene in that particular pathway and test those for antiviral activity. We've done that successfully for other respiratory viruses, and that works in process now for COVID. But it's a relatively new approach to think about identifying cellular targets as a way of inhibiting the replication of a pathogen. And then, of course, we're collaborating with John, not only in hepatitis B, but on siRNAs for COVID. We have a couple of siRNAs, which in combination seem to be effective at blocking the replication of COVID, at least in cell culture. And so we're doing some additional testing on those and moving that forward towards the clinic. So taking a number of approaches. We're also thinking longer term. There's obviously an urgent need to do something about COVID, but it's the third coronavirus epidemic that we've had recently after SARS and MERS. It won't be the last. And so we are thinking about how to have reagents that more broadly inhibit coronaviruses. Our lead antibody inhibits a family, SARS, COVID, and a number of other strains, but it's not pan-coronavirus. And so we were looking for a limited set of reagents that would provide virtually pan-coronavirus protection. I think that's the longer term goal. So stop there. I think that's a short synopsis of our approach.
Thank you, George. And it's very refreshing to see a high-tech company starting in this space of infectious disease, where we have had lots of, I think, initiative to try and bring more innovation into this space. And, you know, at Pfizer, as science leader for the company, we have a legacy. We're still the world-leading producer of antibiotics, but the amount of novel creative science, whether inside our walls for therapeutics or industry as a whole, has declined, which has, I think, been quite worrisome. So I'm very pleased to see the initiative you are taking, and I think it's so important. And we come to wake up, of course, with the COVID pandemic, but also the rise of multi-resistant bacteria and very difficult to treat fungus. Mm-hmm. One thing when you spoke about antibody effector function, I've noted a really fast-moving field using bifunctional antibody in oncology. And we are participating in highly promising myeloma bifunctional antibodies. And I see other companies such as BMS and J&J, all companies indicating that this bifunctional technology can be very powerful in removing cancer cells by retargeting T cells. Do you see bifunctional antibodies also rising in infectious disease, whether to target phagocytes to bacteria or possibly T cells to eliminate viral containing cells and lower the risk of carriage in the population?
Yeah, great question. Yes, is the short answer to that. Look, the idea of bringing in T cells or natural killer cells or other cells to actually kill the cell to which the antibody is bound A number of approaches to that. You know, the FC modifications I talked about enhance the binding to FC gamma receptors on those cells.
Yeah.
Much stronger binding, eliminate the binding to the negatively regulatory receptors. So that's one approach. Bifunctional antibodies, absolutely another approach. It was an interesting observation made by Antonio Lancevecchia, who's working with us now on finding, let's say, transcripts from other regions of the genome naturally inserted into antibodies. And so working on malaria, for example, we had antibodies that were especially potent when we sequenced them. We found the malaria receptor integrated into the antibody. That was a naturally occurring event. That was not a bifunctional antibody that we made. But we've now learned how to harness that system so we can make bifunctional antibodies that have a natural structure. There are many other structures. And so, yes, we are taking those approaches. They may be particularly interesting for bacterial infections for a number of reasons. So yes, is the short answer to the question.
Thank you. That was fascinating. And listening to malaria, of course, I can't resist to discuss now with Penny here. You're leading the largest force in the philanthropic funded impact on global health. I know you're a physician by training, so I'm eager to listen how you see the future of your group when it comes to global impact. I know you've had initially a big emphasis on communicable disease, the most vulnerable, the newborn, the mothers, and of course, many of the solutions has initially been those that are easy to deploy in a, let's say, developing world. And how do you feel that you can tap into this new next generation that we hear about in this panel, from RNAi till highly engineered antibodies to oral peptides and to vaccines, some of them in infectious disease, but others addressing pandemics of different type, like Matt spoke about diabetes and obesity that unfortunately is a pandemic even in developing world. So I'm eager to listen to your thoughts and guidance for the future.
So thank you, and thanks to the organizers for inviting me to participate in this panel. You asked a fascinating question, Michael. You know, the Gates Medical Research Institute, we are a nonprofit biotech. We're a subsidiary of the foundation, and we're working to develop products that disproportionately affect the poor. So we're focused specifically on interventions against tuberculosis, malaria, enteric diseases, and disorders that affect mothers and their newborns, as you said. And you know, a basic belief of the foundation is that all lives have equal value. And in practice, what does that mean? I think that means applying the same cutting edge technologies that we're using in high income countries for oncology or genetic disorders, applying those to the diseases of the poorest that are prevalent in low resource settings. So our disease areas that we're working on aren't going to change. And so that means we are partnering with various entities, be they academic, biotech, pharma, et cetera, to try to find solutions. So let's take TB for an example. TB kills 1.5 million people every year. That is, of course, more than the total that have been deaths that have occurred from COVID to date. And unfortunately, in low resource settings, the population that it hits is young adults, right when they're at the prime of their lives, the most productive members of society. So it's a big drag on economies, et cetera. So if we can end the TB epidemic, those countries their economies can emerge, and the next generation can have a much better opportunity to have a healthy and productive life. So how are we doing that? We're looking at a combination for TB of vaccines and drugs. We have vaccines. We have one vaccine that's a prophylactic. We have another that is actually intended to prevent progression of disease in those already infected. We in-licensed that vaccine from GlaxoSmithKline, and the data were quite favorable for preventing that progression in those already infected to pulmonary disease. So we're excited. We got another phase 2 trial in HIV infected individuals. You know, the burden of TB is very high there. That's ready to start imminently. We're also looking at drugs as well. Until we can get a vaccine, can a novel drug regimen be an answer? The current TB drug regimens are, 4 drugs. You have to take them for 6 to 18 months, depending on the resistance profile of the mycobacterium. We are looking for a safer, simpler, shorter regimen. So we developed a consortium that before COVID, it was considered novel with Evotech, Johnson & Johnson, GlaxoSmithKline, and Otsuka and the foundation to actually look at different novel TB drugs that are emerging from each of the different entities' pipelines, putting those together in novel regimens and moving forward with those to look at efficacy. We're using technologies that have been made available through partnerships broadly to talk about monoclonals. George was talking about monoclonals. We're looking at monoclonals potentially for malaria prophylaxis with the modifications that have been made to extend half-life. Is it possible that we could use malaria monoclonals for chemoprophylaxis before the rainy season occurs? So, all of the individuals in a village get a shot of monoclonal that will protect them through the rainy season. And could that be a bridge until we develop a safe and effective vaccine with a high enough magnitude of efficacy and long enough duration of efficacy to really achieve our goal of malaria eradication? But you ask about non-communicable diseases, and when you look at the literature from WHO, out of the 10 leading causes of death in low-income countries, 6 of the 10 are communicable diseases that can likely be prevented through vaccination. So that's where our focus is. But we also realize when you look at the epidemiology that chronic diseases such as cardiovascular disease, cancer, diabetes, and respiratory diseases are increasing in low-income countries. So what will be the solution to those as communicable diseases are addressed? And I was thinking about this. I think what we need to do is really think about innovation happening at the intersection of different disciplines. And what do I mean by that? I mean, if you look at COVID vaccines right now, the adenovirus vectors, they had their origins in gene therapy. The nucleic acid vaccines really were thought of as an approach for oncology, for cancer. So, could it be that, and I think those are wonderful platforms. Yes, they need to be optimized for low-income countries, but I think there are wonderful platforms that can be applied to the diseases I just talked about, whether it's TB, malaria, enteric diseases, et cetera. Optimization and formulations, we certainly need that. They're stored frozen over the long term. We need better stability of the vaccines, but I think that they could certainly be possibilities for vaccines against communicable diseases. Paul Stoppel's J&J group, they have their adenovirus 26 vector-based HIV vaccine that's in late-stage trials, and we're really excited about the potential of those results. But I started to think about, okay, if my dream is that 2030, 2035, we will have vaccines for malaria, TB, HIV. So how will we address these non-communicable diseases? And I was just thinking, well, could we do the reverse of what we've done in high-income countries? So we've brought disease technologies from other areas and applied them to COVID. Can we take the technologies that we are currently going to be using for communicable diseases in low-income countries and go back to the beginning again and utilize them to address some of these non-communicable diseases? Is it possible, I don't know, that gene editing in the future could actually be like a vaccine, say, for cardiovascular disease for a low-income country. So I think the possibility is there, and I'm excited about it. First, we have to focus on preventing these 6 leading causes of death that are caused by communicable diseases.
Yeah, no, that makes a lot of sense to start in low-hanging fruit and the difficulty of not using in the best way available treatment for some of these devastating infectious disease. On the other hand, you know, looking at the rise of medical healthcare in Asia and China particularly, from being not that many decades ago a rather economically poorly developed to now being one of the biggest contributor in science technology. Just looking at COVID, I've been amazed looking at just the public open literature databases, medRxiv, bioRxiv, the science from China. Obviously, they got them months ahead of the Western groups since the COVID pandemic started there. But the number of great papers are just amazing. So that nation has been turned from being at the lower tier of innovation to a driver of new innovation. So as you mentioned gene editing, it strikes me that many of the diseases that today could be treated with gene therapy or in the future, the technology isn't at all available, I think, in Africa. And if you take a disease like hemophilia, certainly not at the top of the number of impact on mortality in Africa, but in the quite close future, we will be able to have a single infusion of gene therapy to cure, or at least give 10 years of free of hemophilia treatment. And I do assume that in Africa today, most patients with hemophilia probably don't live a very long life because of the difficulty to get access to weekly treatment in the IV. So I was just thinking if some kind of a subsidiary of the Medical Research Institute of Gate picking up a new technology could lead to a whole new generation of physicians and scientists that would drive those type of technologies into all kind of treatment options also in the developing world.
Yes, absolutely. And in fact, this is an area the NIH and the foundation is collaborating on right now to look at what could be the possibilities specifically with sickle cell disease. And I think you're exactly right. Another way that the Institute and the NIH is collaborating is through fellowships for African scientists. It's so important that we support scientists in Africa and continue to help them advance their labs and advance their discovery science as well as development. And so that's something the NIH has a fellowship. And so once the fellows rotate through and finish their fellowship at the NIH, then they come to the Institute to actually learn more about product development in addition to discovery science. So I think this is how we build that foundation so that in the future, as new technologies like gene editing, et cetera, become more optimal and better characterized, that it could be possible that they could become solutions. Things that we just wouldn't dream of now being solutions for low-income countries could become solutions.
That's great to hear. And that dream that you spoke that isn't here yet, I'll take the opportunity to share with you that a few of the gene therapy pharma companies are in discussion with NIH and FDA to create for the very few in the world, those that suffer from ultra-orphan diseases, to create a shared institute on philanthropic base with a view when you save one life, you show the globe that every life matters. And maybe it's an opportunity for your institute to join with a view of opening up those technologies, because we will do them on a more simplified way to quicker adapt to maybe 100 patients in the world suffering from a disease. But maybe we could use that kind of philanthropic gene therapy institute to also make some of your dreams come through. Now let's take an opportunity. It wouldn't be a great panel on the latest days of October if we didn't speak a little bit about the pandemic. So I'm going to go back to Mads Krogsgaard here. In my conversation with you, I learned that your company is actually also active in thinking about medicines that can be helpful for the pandemic. And I think some of the data you will discuss with us are, I think for many of us, data we didn't think about or are unaware of, addressing a major risk factor in the pandemic related to metabolic dysfunction and obesity.
Yeah, Michael, it is true, as you know, and as you explained, that the major risk factors for poor outcomes in COVID-19 patients include obesity, diabetes, hypertension, old age, all these, you can say, unhealthy lifespan elements that unfortunately come together and greatly increase the morbidity and mortality among such people with COVID-19. So we are lucky enough to have contributed to an ongoing collaboration with NIH and other parties in the US to really interrogate the databases that exist. And there's a cohort called the N3C cohort within NIH, where it's been possible basically to look at what are the outcomes among around 1 million or so lives in the US and literally dozens of thousands of COVID-19 victims. And the early findings that, of course, we need now to confirm, and they will be published very soon, I believe, they actually suggest that there are really big differences between the classes of anti-diabetic medications and the newer ones that are also cardioprotective, such as the GLP-1 receptor agonist. They are apparently showing up with mortality reductions in the realm of what you would expect to see with remdesivir and dexamethasone and so on. So apparently, even when you go into non-COVID-19 related treatment modalities, there is a huge difference on the impact of the individual of what kind of treatment they're taking for their disease, in particular if it's a disease like diabetes and obesity, which really predisposes to poor outcomes. So we're very excited about that. And of course, it also signals that, as has been mentioned earlier on, with coronaviruses apparently continuing to come back in new shapes and forms and creating pandemics, I think we need to see some of these chronic non-communicable diseases as ones that are making people also very susceptible to poor outcomes for the communicable diseases. So there's an interesting interplay here that we'll learn much more about, and these early data, I'm sure, will be made available very soon and very interesting.
Thank you. Now we'll turn to Jeff Spaeder at IQVIA. Please share with us how your company contributes to the advance of science, medicine, and healthcare, and also particularly what you have learned using your digital technologies in this early 7-8 months of the pandemic, or
Sure.
10 now.
Yeah, no, it's interesting, Michael. First of all, thank you for hosting this. This is obviously not how any of us expected this panel to go, and to be able to pull this off remotely with people literally all over the world. Thank you for taking that on. Yeah, so IQVIA, I think, had some interesting insights into this. We actually have a large presence in China. And so when the outbreak started in Wuhan in late 2019, it was affecting some of our people in our studies there. So in some ways, given our experience and our footprint in China, we had a little bit of a head start on seeing what was happening. And interestingly, it's, as China has recovered, it provides, I think, a little bit of a perspective of what the rest of the world may go through. So we're entering this almost a year now of dealing with this. I think a couple observations from our experience is our company was brought together from the legacy quintiles and IMS health organizations about 4 years ago. And the thought was, how do you bring digital medicine and data and analytics, and how do you bring them together, and what's the value of them? And early on in the in kind of the due diligence, as both organizations were kind of assessing this partnership, it became apparent that having data in and of itself was not sufficient to make insights. You had to really combine that with the other components of digital health. You had to have transformative technology to be able to manage these large amounts of data. You had to have the analytics to be able to manage and gain insights out of the data. And you had to have pretty broad and deep therapeutic and domain expertise to ask the right questions and interpret the things correctly. And so, you know, we've brought our experience is that you have to have all four: data, technology, analytics, and domain expertise in order to effectively work in digital health. We have seen over, you know, prior to COVID, it was a lot of interest in doing digital medicine. The many sponsors and healthcare organizations wanted to do it, but there was just an inertia to prevent the ability to actually do it. And COVID essentially swept away, almost like a tsunami, a lot of those obstacles. It was, you know, in terms of chemistry, it was almost activating energy. And you know, all of the obstacles that you had in the past to say, well, we don't want to do this, it's new, how's the reimbursement going to work? You know, all of those things were swept away, and it really focused, you know, people on the commercial side and on the R&D side to really look at things in a new way and to really embrace things. And so, whereas a year ago, there was always maybe tepid interest in thinking about digital health, now, both on the commercial and also the R&D side, lots of interest. And I think it's a lot of interest really driven by three things. The first is the need just to adapt to this environment where the COVID outbreak and lockdowns had a significant impact on just the way we work on a day-to-day basis. You had to navigate around those obstacles. Secondly, I think there's a realization, and George, you had mentioned this. You know, this is the third coronavirus outbreak that has had significant morbidity and mortality in the past 2 decades. It is likely to happen again. And so I think many organizations are thinking, if it's happened once, it's likely to happen again at probably the most inopportune time. So we have to think about being able to interact with patients and subjects in clinical studies in a different way, and essentially to reduce the risk. And then I think the third thing also is this outbreak has really highlighted disproportionate impact of the disease on underserved communities. I think we've always known that, but it's really highlighted this in a way that is quite frankly very stark. And I think there is this thought about digital health that is maybe more egalitarian in terms of how you can reach out to communities, especially those that are underserved, and in some ways undoes some of the conscious or unintentional systems that are in place that may exacerbate and continue some of those inequities and inequalities. So I think there is this greater appreciation of the need to work in underserved communities, and you know, there are obviously some innovative things, and you know, Penny was talking about that on a global perspective, but technology and a digital outreach is probably going to be a component of that to break down some of these barriers to treating traditionally underserved populations.
Thank you very much. I see we are starting to get a few questions, and we will soon start to address those. And I just encourage other participants listening in to the panel, there is still room to submit more questions. We have not forgotten you. Staying on the topic of pandemic, I would be keen to return to John at Alnylam here. I know from our earlier conversation with you and George that you are exploring the RNA technology also for COVID. It started to be a very crowded space, and your technology has been so successful in targeting very well validated gene targets, gene expressed RNA targets in human disease. Now, what made you feel that targeting viral proteins at the mRNA level would be worth while to add, given what we see, many antivirals, many antibodies, we heard comorbid conditions being treated from meds. How do you see your approach being differentiated here?
Well, I mean, for starters, Michael, as you know, we still have, we have many shots on goal for developing treatments for SARS-CoV-2 and COVID-19. You know, we still don't have clear winners. And so I think it is important that the industry marshal its resources, its technologies in the fight against this pandemic. I think, frankly, I view it as a moral responsibility at the very least. And given the fact that RNA interference targets RNA and the fact that SARS-CoV-2 is an RNA virus, there's certainly a very strong logic structure to look at this approach for developing an antiviral. Now, obviously, you know, we have to deliver it through inhalation, which is different than what a small molecule could do, but it would be less invasive than an intravenous infusion using a nebulizer, for example. So there are some benefits. I think where there could be some interesting opportunities, given the long durability of the RNAi mechanism of action, is to consider it in a prophylactic mode where you could administer it relatively infrequent to at-risk populations and achieve some attenuation of an infection. And so this would be orthogonal to what you could achieve with you know, antibodies or with a vaccine-based approach. And it's really representative of how I believe strongly the industry must marshal its broad technology opportunities to fight this public health crisis that we're all facing right now. Now, in addition to targeting SARS-CoV-2, earlier in our efforts, we're also interested to explore host factors. And we do have an effort looking at ACE2 as a target, as well as TMPRSS2, which is another well-defined host factor target for SARS-CoV-2 replication in cells. And so we'll see how those look as well as potential alternative targets from the virus itself. But that's the effort, Michael.
So we're very grateful for marshalling your resources, and you have a very impressive track record to build on. I can't resist intriguing to hear that you are considering inhalation.
Correct.
Does that mean, because you have a good track record, I think, with both IV and subcutaneous for systemic delivery, does that mean that you want to reach higher RNAi concentration in the airway epithelial cells, or you even think that you will penetrate circulating viruses?
Well, the data we have shows that the best knockdown of target genes in the lung airways is through an inhalational route of delivery. You do get some effects if you give it systemically, sub-Q or IV, but not as efficient, not as well as if you give it through an inhalational route of administration. And obviously for a respiratory virus like this, getting good concentrations into the lung airways is going to be important for maximizing efficacy.
That's great to hear. And I think we've heard there are some inhalation studies going on with interferons too. So maybe this is a whole space that you're opening up for viral airway diseases. One of the questions that came in here, if the pandemic second wave and future waves are modified COVID virus, how are we addressed being ready to tackle that next year or after? And maybe that was George that created that fear factor and eliciting questions. George, anything in general you think the biomedical community are doing that's more concerted than after the SARS, and what you are specifically thinking to be prepared for the next wave?
Look, I don't want to be alarmist, but it is a serious issue. there are variants of COVID now that are arising that are probably the first truly immune evasive mutants. So you may know that SARS and COVID both bind to the ACE2 receptor. They bind to the same receptor. In both cases, the main binding energy comes from a salt bridge, but the salt bridges are different, right? So they use different amino acids on both the receptor and the virus. There's a mutation now that has arisen in Scotland, a variant, which has both salt bridges. And that is resistant to some of the antibodies that are now in development. So there has not yet been selection on a broad basis for viral strains that can evade the current therapy. So I think there will be for sure. And so we're going to need collections of antibodies, each of which are refractory and each of which bind to conserved epitopes, each of which is more difficult for the virus to evade than the current generations of antibodies over the long term. Right? This is not something that's going to happen overnight. And so the current generations, I think we should think about and be as something that can help us in the near term, certainly are likely to be effective, that eventually there'll be resistant viral strains and we'll have to bring forward the second, third generation products. And as long as we keep that in mind and are working on them, we'll be okay. But to ignore it, I think we'll regret that.
Thank you. And I know from the various companies participating in small molecule antivirals, kind of a traditional and well-established modality, just to give some calm to the participant and ask the question that many of those, whether polymerase inhibitor or protease inhibitor, show features of being quite active across all known coronavirus. The challenge is that you need to treat early and you need to establish cocktails of medicines, which has been the way we, as a biomedical community, have shown cure-like effects for HCV and chronic management of disease for HIV. So we need to find a way to create cross-company partnership in looking at combinations so we're prepared. And you could, of course, think about an effort where you create, if you use protease as an example, a library of proteases and map which drugs are active. So when we're hit by a new coronavirus, we already have a digital database to go to. But unfortunately, on the other hand, preparedness for the future hasn't been a great human skill. We seem to be often very much occupied with immediate near term. Another question here was, do we know whether cumulative vaccination for different diseases does not cause damage to our body? Maybe I'll ask Penny as a neutral, independent voice and not representing any company, if you have any advice to share. I guess this is a colleague that are kind of considering, is there any need in this public health education to be concerned about too many vaccines, because we have a lot of anti-vaccination forces out there too. Penny.
Yes. So thank you for this question. And it's a question that I often heard from parents back in the day when I was practicing. And in fact, from the time an infant is born and their trip home, the first time they go out of the house, they're exposed to many different antigens, many more antigens than are in vaccines. So while this is a common worry and something that anti-vaccination organizations bring up, in fact, the science has not borne that out. There are multiple databases that the CDC has where they follow the safety of vaccines over time in large populations. When I say large, millions of children and adults, and look at safety over time. And there is absolutely zero evidence that multiple vaccines, that there's some kind of cumulative harmful effects to the body. And in fact, you know, it's exactly the opposite. In fact, we know that the benefit of vaccines is enormous. I mean, when you look at the benefit of vaccines, you look at the early 1900s as compared to now. Look at you know, childhood deaths from diphtheria, pertussis, tetanus, you know, rotavirus, pneumonia, pneumococcal pneumonia, et cetera, and how those rates have just plummeted in the US to near zero. Measles was at near zero before, you know, the recent outbreaks last year. So the benefit of vaccines is unassailable. It's there, and the safety has been monitored. This is not a worry.
Hey, and Penny, it's Jeff. I would actually say if you want to look at a non-vaccination world, you look at COVID. Right. I mean, this is exactly what happens when you don't have vaccines, and you can see what the impact is on that. So I think you're absolutely right. The amount of antigen epitopes you get just every day far exceeds what happens, and I don't think anybody wants to see COVID again occurring. But that's what happens if you don't have vaccines on a societal basis.
Thank you. Another question is, for all the panelists focused on SARS-CoV-2 prevention treatment modalities, what are the most promising approaches to provide continued efficacy in the face of mutation of the pathogen to more lethal genetic variants? So maybe we could ask Jeff to share, do you think we have enough of a concerted digital effort to monitor all those genetic sequences and be able to see if there is an outbreak of a variant causing a different characterization of the disease, or are we just chasing case reports in Lancet and New England Journal of Medicine?
Well, I think this is really highlighted, Michael. I think it's a great observation. You know, there has been, you know, aside from relatively small numbers of organizations, you know, and some of those people are actually on this panel right now. Until recently, there's been almost a hubris about communicable diseases, that it was something that happened, you know, to our great grandparents, and it's not something we really have to worry about now. And you know, that type of lack of humility, and nature has a wonderful way of enforcing humility upon us, and it, I think, highlights a couple things. One is the public health infrastructure is so critical. We don't think about it in the setting of acute illness, but this is just, again, highlighted the importance of it. And you look at some of the countries that have been able to navigate this extraordinarily well. So you look at Taiwan as an example. The ability to integrate in a public health response very effectively has allowed some countries to be able to navigate this outbreak extraordinarily well. And at the center of public health is digital tracking. It's the ability to track longitudinally what's happening with individuals, with patients, and that digital infrastructure requires scalability, interoperability, and interchangeability of data. And you know, you talk to some of the people that were involved in the HITECH Act back in 2009 that incentivized healthcare providers to move into electronic health records. There was always this assumption that the first decade was going to be just putting the infrastructure in place, and that it really wasn't going to be until 2020 to 2030 that you'd really be able to see the benefits of that digital health infrastructure. And I think this has been a huge wake-up call that we have to continue to invest in that infrastructure to be able to use electronic health records and, frankly, all sorts of data. You know, we have this phrase at our company, human data sciences, because it's, you know, we specifically said it wasn't medical data sciences is the important thing. There are so many things that have a huge impact upon health, independent of your medical status, of your genomics, where you live, your access to healthcare, your access to food. Like, these are all things that make an individual uniquely human and makes some particularly susceptible to disease and others more resistant. So I do think this has been a wake-up call. There has got to be a better public health infrastructure. The interoperability and accessibility of health data is critical. And then, I think one of the other things outside of coronavirus, if you're looking at this and if you're suffering from acute myelogenous leukemia or pancreatic cancer or sickle cell disease, you look at the speed with which therapies and vaccines have been developed for coronavirus. And I think you have to ask yourself, why not me? I definitely am going to have a bad outcome of this. And the ability to look at long-term efficacy and tolerability and the way that we change how we do clinical research, my hope is that it allows us to accelerate the development of therapies, get them in the hands in a risk stratified type of process to patients that need them the best, and then be able to collect more and more information in the real world and from the digital health infrastructure to help to refine some of these decisions, because more people die of malaria or tuberculosis every year than what's happened with COVID. And I think we have to take some of the learnings from COVID and apply it to other areas as well.
Thank you. Another question here with a historical perspective. How can we apply what we have learned from Spanish flu, I guess that was 100 years ago, that we can apply to COVID-19 treatment? George, any advice from your side?
Well, social distancing works. I think that's the first thing to apply. Look, Michael, I think all these years later, 100 years after Spanish flu, we do have some small molecule drugs against flu, neuraminidase inhibitors. Flu does have the ability to escape them, right? So there are resistant strains of flu now emerging. So the challenge is really to find drugs, as you alluded to, which have a very high barrier for the virus to escape. Small molecules can do that, and certainly do that for HIV. I think antibodies also can do that. And if you think about the immunodominant epitopes of a pathogen are almost by definition ones that the virus doesn't care about, because there's been no selective pressure against making them non-immunodominant. So the virus is waving these things out in the air saying, look over here, and the immune system generates reagents against those epitopes. Things that are crucial for the virus, things that are essential for the virus that it can't change, are hidden. They're not waving out. Viruses that had them accessible aren't around anymore. But antibodies do exist against those. They're rare. You have to look for them. You have to have the right screening assays. You have to have the right technology. You have to have the right skill set. So in the case of flu, we have an antibody that recognizes every strain of flu A that's arisen in the past 100, potently neutralizes. So the 1918 pandemic strain and every strain that's arisen since then, seasonal strains and pandemic strains. For Ebola, we have an antibody. We may have one like that now for COVID, time will tell. So there are ways to attack the current pandemic that are likely to be durable and be very difficult for the strains to escape. And the antibodies might be one way, small molecules are another, combinations of SIs could be another. And so I think at this point, for COVID, we need to try them all.
Another question here, how many mutations of SARS-CoV-2 do we know of today? And the current vaccines, can they deal with those mutations? I would provide one quick answer, and I encourage anyone that wants to pitch in. Most of the vaccines, or almost all that are advanced preclinical to in the early to mid to late clinical, most of them focus on the spike protein, as you know, the docking protein of the virus with a receptor ACE2. I think John spoke about that too. And most of the efforts have been to stabilize the spike in its most active conformation. And those vaccines that have it in the active conformation seems, to the best of my knowledge, be not yet addressed by any evolving mutants. And probably that reflects a bit what George alluded to, that that's such a crucial protein in the virus that it will take a number of generations exchange of genetic material between different coronavirus to create a strong enough new entry receptor or reorganize the spike protein. I think there are some vaccines that have the spike protein in a more neutral position and may not equally well open up the most crucial epitopes that the virus seems yet not to have mutated. I think we saw some signs that once the spike is cleaved, it opens up additional binding to a peptide that's involved on airway cells as an entry receptor in addition to spike ACE2, I think. So clearly there is more to be learned, but I think the beauty of vaccines are that they're polyclonal, bringing back to the discussion that the importance of often having a collection of antibodies. And so at least for, I'm hoping, the next couple of years, these vaccines will be still active. I think the threat is a lot of stuff is going on in the animal kingdom, and that can lead quickly to new coronaviruses to emerge. I'll ask John if you have any other thinking on the work you are doing, how to deal with this risk of mutations that may make your RNA weakened, or whether you feel that you have a few years to go before that may happen.
Well, you know, it's interesting. When we began to look at sequences coming out of the initial isolates from China, we were quite impressed with the conservation at the RNA level of these viral isolates, all the way back to the original 2003 SARS and MERS even, although MERS is a little bit more divergent, and then other coronaviruses that normally circulate in the population. So the sequence conservation was really impressive to us, and it gave us some encouragement around being able to even use one single sRNA for this virus. But we soon learned that we have to use two, that you can generate resistance, again, collaboratively with George's group, you can generate resistance. And so having two sRNAs toward highly conserved sites allows you to avoid that issue. But I think in general, what we don't yet know, Michael, is how the virus may, over time, mutate under selective pressure. And with the advent of new therapies, treatments, small molecules, antibodies, RNAi, vaccines, we have to keep our eyes out for this issue. And I don't think it's out of the question that we'll need to think about evolution of our modalities over time to address future mutations that emerge that require adjustments of our modalities to address them. I don't know, George, if you have a view on that as well.
No, agree completely, John. We'll see. I think Michael's point is true, that I think we'll be able to contain COVID-19, and I think the biggest danger is additional coronaviruses that come down the pike. This is highly unlikely that it's 3 and out, and we won't see any more. How related they are, whether or not the current reagents will work against them, is completely open question. So I think as we think about, as you said, the sequences that are currently conserved among different coronaviruses, those would be, I think, a really good place to focus, just because there's a higher chance they'll also be conserved in the next coronavirus.
Yeah, it's fascinating to see the role certain unusual animal species have played here. Bats that have evolved unique symbiosis with coronavirus and carry so many of them. And I just learned that there was a very powerful bat immunology society that has been surviving for many decades, but the funding to it was just disrupted, I think, 2019. So hopefully that can be restored, because monitoring the evolution of coronavirus in bats is likely going to give us, as George spoke about, how these swaps between viruses of genetic material that they can do may then lead to viruses that can cross species barriers. And when they can cross more than one species barriers, it's probably a good alert signal about the risk in a few years for humans. We spoke about numerous modalities here, and of course COVID-19 shaped a lot of our questions, but I thought I'll ask Mads a bit, what do you think about the future of modalities aimed to treat degeneration of organs, degeneration over the aging when it comes to organ health? And there's been a number of efforts in trying to understand the biology of aging and the ability to rejuvenate cells, including working on regenerative medicines using cell-based approaches, or trying to reconfigure the gene expression patterns of old cells to new cells. And I think there are even some examples now, quite fascinating, of transferring soluble factors from young animal to old animal and see how you can change gene expression. Is this an area your company has been starting to have interest in, regenerative medicine, as you focus on some of the communicable diseases that seems to be, by increasing age, causing a growing contribution to mortality and morbidity?
Yeah, I think it's really interesting, Michael, that we actually started out of a basic research interest about 25 years ago, working on developmental biology, trying to understand what happens in utero during the first trimester of a pregnancy, and how can we replicate that to recreate cells that are either died or dormant or whatever in certain degenerative diseases. And interestingly, as we see now, is the emergence of a phenomenal understanding of exactly what triggers the omni or totipotent stem cell to go down one ecto, endo, or mesodermal pathway versus the other, etc. And once you harness this understanding, it actually turns out to be pretty easy within a matter of a fortnight or so to differentiate any kind of cell type within the human body. So right now, our belief is that there are some very, very difficult diseases to treat, and one of those is, of course, type 1 diabetes, because whenever you implant a new beta cell creating insulin production, you also have to shield it from the immune system. You have to oxygenate it because it is extremely oxygen demanding. So there are some areas, such as autoimmune diseases with high consuming cells that are very demanding to treat with a regenerative medicines approach. And then there are others where I have to say, based on the animal studies we have done across different disease areas, where it actually seems to be pretty promising and straightforward. So for instance, if you take Parkinson's disease, in most cases, it's a highly localized disease with the death of the so-called dopaminergic neurons. And if you are able to either in one or two operative, you can say, implantations, recreate that which has been lost. I think we know from, among other places, Sweden, where these experiments have been ongoing with fetal implants 20 years ago, that it can indeed work. And our job, what I believe will make a big difference, is if you can have a number of, you can say, GMP compliant stem cell lines with different haplotypes and so on, that you can then differentiate basically upon demand at large scale into the target cells of desire. You can freeze them down. There's high viability, maybe 8% viability after thawing, and have them shipped directly to the center where the implantation takes place. This is a very different approach from classical pharmacotherapy, because the collaborators you are collaborating with are very often specialized surgeons and not the classic endocrinologist or oncologist that one is used to collaborating with. But I truly believe that in the next 5-10 years, we're going to see a lot of neuroregenerative medicines, and some of them are more scalable than CAR-T therapies, where you have to use the individual's own cells as a basis for a factory. In this case, it could be scalable to the extent that one bioreactor could potentially lend itself to treat literally thousands of people with some kind of degenerative condition.
Thank you. When you work with regenerative medicine, as you describe it, I think there are a number of key gene expressed pathways that needs to be modulated. Maybe also epigenetic rewriting can be important. So I was thinking, John, you have likely access to one of the most powerful libraries or ability to design libraries to deal with some of these factors that determine cell aging, cell senescence. Do you have any garage research that you are open to share, screening using the RNA powerful technology for cellular age and ability to be more long-lasting or have a younger phenotype?
Great question. Well, I mean, Michael, we've left the use of our technology for, genetic validation of targets to the academic community. That's where that great research often takes place. And we've just kept our focus and our resources on making medicines very directly. So, aside from some work we do with human genetic databases, together with you, as you know, the UK Biobank and other efforts, and Regeneron, we're very focused mostly on translational research, not on the basic elements that type of work would entail.
Do you think there are any clues for using the type of RNA vectors? There are some genetic diseases that show accelerated aging, and you do have increasing, I think, exciting research around the centenarian, a community of people that do really well and are above 100 years. There are not many of them. So, of course, a small patient pool. But do you see new leads in more of these genetic disease, not related to disease, but more to intermediary phenotype that could open up this space of long-lasting medicine?
I absolutely believe in those opportunities, Michael. And there are many interesting targets, again, mostly from the work we've been doing with the UK Biobank to identify, interesting loss of function traits or mutations that could help us define medicines that address, larger unmet needs beyond the rare disease setting that we've initiated with. One similar example, it's a little bit different, is what we're doing in hypertension, trying to target angiotensinogen with RNAi therapeutics as a way of reimagining the treatment of hypertension, which of course is probably the leading cause of cardiovascular morbidity and mortality in the world, even more than LDL cholesterol. So that's an area of great interest to us that is advancing into phase 2 clinical trials next year.
That's great news to hear. One question that I think is fit for George here, from the participants, do we have to worry about antibody-dependent enhancement in COV-2 antibody treatment or vaccines with additional mutations? And maybe this links whether the virus will be able to evolve to make some of the principles intended to protect us actually to deteriorate our treatment outcome. Do you see a risk for that, George, or you think we are enough screening for that and can write it off?
Well, there's no sign of antibody-dependent enhancement in COVID so far. I think the 100,000 patients or so who've gotten convalescent plasma which is a mixture of all kinds of antibodies, have no sign of any antibody-dependent enhancement. One of the two principal routes of antibody-dependent enhancement is the Fc receptors on antibodies dragging the virus into myeloid cells where the virus can replicate, not only kill the cells that would kill it, but have another reservoir in which to replicate. COVID does not seem to replicate in the myeloid cells. So that mechanism of ADE probably is not operative in COVID. The other potential one is complement fixation, which sometimes can enhance diseases. I think that is still somewhat of an open question. There's no evidence of it, but I don't think it's been definitively ruled out. We have, one of the Fc modifications we made to enhance binding to Fc gamma receptors at the same time eliminates complement fixation. So we'll see. I think these are good questions, good considerations. I don't think there's any evidence yet to support ADE. Clearly, there's no replication in myeloid cells. And so I think it's a minor concern, but something we, of course, have to keep an eye on.
Thank you. Making sure at the very last minutes here that we end with a note on focus on the almost 8 billion people and not just the sophisticated medical care in the developed world. Going back to Penny here, when you look where we are now, 10, 11 months into the pandemic, and we hear about early promise of certain antivirals, one emergency use authorization and now approved in US. We hear about some impact on monoclonal antibodies. We're waiting eagerly over the next weeks to months to see the outcome of vaccines. We hear about inhaled RNAi coming from John and George here. And of course, other opportunities that we didn't touch upon for SARS-CoV-2. Are you feeling that you are reasonably satisfied with how the rollout of those, fingers crossed, successful modalities will benefit in a fair, equitable manner the global population? Where do you see the biggest bottlenecks for you to feel really good about that we won the battle against this disease, maybe 2022, reasonably fair and equitable across the globe? And are there any call to action you would like to do to make sure that we do better, Penny?
Yes, so thank you for that question. I'm really excited about the vaccines. They've been developed at unprecedented pace, at 1/10 of the normal pace. And I am cautiously optimistic that the data will be favorable. The Coalition for Epidemic Preparedness Innovations is supporting the research and development arm of the ACT Accelerator, working with Gavi and WHO to try to ensure that vaccines will be distributed equitably around the globe. And I think that effort has a tremendous amount of momentum and strength behind it with, over 160 countries now being involved. So I'm very encouraged by that. I'm less encouraged on the therapeutics side. The monoclonals are promising, but are we going to have the capacity and that's needed to actually provide them not only to the rich world, but also to low-income countries? Of course, Foundation, we have partnerships on that. We've reserved, priceless capacity for providing those monoclonals to low-income countries, but I think there's more to be done there. And with the antivirals, there are a few promising things on the scene, but we need to be much better prepared the next time around to move more quickly with the antivirals so that they can serve properly as that bridge until monoclonals and vaccines are available. And I guess I would like to end by saying, we have a couple of great tools right now that we all need to be using. And I know I'm preaching to the choir here, but wear your mask, social distance, wash your hands, because we can do a lot to dampen the peak of the second wave right now with those simple public health tools.
Thank you very much. We are coming to an end here. Our big thank you to the panel for sharing your time here with us, to Galien and Bruno Cohen for continuing to organize this fantastic learning opportunities for all of us. And I know that you're also planning to go to Africa, which is with a Galien, certainly a good spirit to ensure equity and access to learning about breakthrough medicines. I hope everyone been listening in got a sense for biomedical innovators across all organizations are doing in 10 months what usually takes 10 years. So a big thank you for all of you that have shown the generosity in your time and resources to make a difference here. Have a great day.
Thank you.
Thank you, Michael.
Thank you.
Thank you.
Thank you.